Welcome to the Chapter 5, Part 2 lecture on proteins. You should use the information in this lecture to complete the Chapter 5, Part 2 guided notes, which of course you should complete before you attend class. In this lecture, we're going to talk all about proteins. We're going to talk about what they do, how they're put together, and how they're folded into a three-dimensional shape. We'll also discuss how things like mutations can affect the shapes and functions of proteins. So let's get started. So, what are proteins and what do they do? Well, most people know that proteins are things that you get in your diet from eating things like meats and fish, eggs, and legumes. And they know that they're good for building muscle, but not really anything beyond that. Well, this is what proteins look like in reality. Proteins are big, complex molecules made of hundreds or even thousands of little subunits. They have many different shapes and sizes. There are hundreds of different proteins inside your cells, and they all have different jobs. So proteins are complicated. If you've ever wondered what your DNA actually does, well, what your DNA does is it actually holds the code for making proteins. So the DNA stores the information for making these molecules, and then these molecules actually do all the work of the cell. So by controlling the DNA, really you're controlling the proteins. So proteins are extremely abundant in every single type of cell. If you took a cell and you dried it out, you dehydrated it, you'd find that 50% of the dry stuff that was left over was made only of protein. What kind of things do proteins do? Well, they do all of these things. They do lots of jobs. Again, they run the cell. So if you imagine a cell as being like a little city, the proteins are like the little people inside that do all the little jobs. So let's go through some of these. This diagram comes from your textbook, and it illustrates just a few of the functions that proteins perform. One of the most important types of proteins in your cell are these things called enzymes. The enzyme is this purple blobby thing, and that's its particular shape, and you can see that what it's doing is it's grabbing this dimer and it's breaking it down into individual monomers. So enzymes control chemical reactions inside your cells. They break things down or they build things, and they control the rate at which these things happen. There are many different enzymes inside your cells, and they all have slightly different jobs. We'll talk about them more over in Chapter 8. Some proteins have a protective function. These proteins are called antibodies, and notice that they're shaped like little Ys, which is kind of interesting. But what these things do is they stick to the outside of invading particles like viruses and bacteria, and they help prevent them from spreading. So that's an immune protein. Some proteins act as storehouses for building other proteins. A great example of this is egg white. Egg white consists of a protein known as albumin, and albumin can be broken down into its monomers, which are called amino acids, and that can be used to build other proteins. So you find this albumin inside bird eggs, like chicken eggs, and it's there as a storehouse to build proteins for the little chick that's growing inside that egg. Another type of protein is known as a transport protein. We're going to talk about this in more detail in Chapter 7. Transport proteins are gates. They're built into plasma membranes, the cell membranes of cells, and they help control what goes in and what comes out of the cells. So they're the gatekeepers of the cell. Here's some more functions of proteins. Some proteins help your cells communicate with one another. These include things like hormones and things called receptors. Hormones are proteins that are released in the blood, and they circulate around in your body until they reach what are called target cells. Then they bind with those target cells and they send messages. A great example of a protein hormone is insulin, which is released by the pancreas, and it talks to the liver and your muscles and tells it to remove sugar from the blood. Now, this system wouldn't work without receptors. Receptors are little proteins built into the membranes of cells, and they will connect with the messages, and that causes a chain reaction to happen inside the cell. So they're like the mailboxes that receive the message. Finally, we have two examples here of structural proteins. Some of the most famous structural proteins are found inside your muscles. They're known as actin and myosin. If you could see inside your muscle cells, you'd see that they are just filled with these little proteins, and what these proteins do is actually grab each other. When your muscle contracts, these guys grab each other and pull, and that's what allows your muscle cells to contract and get shorter, and that's what allows you to apply force to something. Here's another example of a structural protein known as collagen. Now, you may have heard of collagen in skin products. Lots of skin products claim to have collagen in them or help build collagen. Look at this protein. It's shaped like a bundle of springs, and being stretchy is essentially its job. Collagen is found deep inside the skin, and it's the protein that allows your skin to stretch and pop back into place. Now, as we get older, our collagen begins to break down, and we don't make as much of it anymore to replace it, so our skin begins to sag and develop wrinkles, things like that, and that's why you see it in a lot of beauty products. Now, beauty products that claim to grow collagen, those might work, depending on what's in there. Beauty products that claim to contain collagen, on the other hand, that's probably not going to help you at all because collagen is a big protein and it can't soak into your skin the way a little bitty molecule can. Right? But those are some functions of proteins. Like all biomolecules, proteins have monomers and dimers that are specific to them. In the protein group, the monomer is known as an amino acid. Here we see two amino acids being put together into the dimer. The dimer of the group is known as a dipeptide, and these amino acids are held together by a peptide bond. Anytime you see that term pep, I want you to think protein, because pep often refers to protein. Now, before you leave the slide, I want you to think, what kind of a reaction is this that I'm seeing? I've got two small subunits being put together into one large subunit, and look what's coming out. What kind of reaction is that? You know this. The polymers in this group all consist of long strings of amino acids held together by peptide bonds. There are two different kinds of polymers that can exist, and these really are put into these categories based on their function. Proteins are fully functional, so they can go out and do a job all by themselves. Polypeptides, on the other hand, are not fully functional by themselves. They have to be combined into larger machines in order to perform their function, in order to do their jobs. Again, the monomers of this group are known as amino acids. There are actually 20 different amino acids that exist. Their names end in "ene." So this, for instance, is tyrosine, this is asparagine, this is valine. One of the skills you will practice in class, and you will be tested on later, is labeling the parts of an amino acid. So let's take a look at it. Even though there are 20 different amino acids, they all have certain parts in common. At the heart of an amino acid, we have this carbon that we call the alpha carbon. Now, because carbon can make 4 covalent bonds, there are going to be 4 things attached to it. All amino acids are going to have a carboxyl group on one side and an amine group on the other side. This is where it gets its name, amino acid. Remember that carboxyls are acids. You will also find a hydrogen attached to that alpha carbon. It's kind of there just as a space filler. And then finally, you'll have something known as the R group. Now remember that R in chemistry is a wild card. It means that there's something attached there, but we don't know what. All 20 of the amino acids have the amine group, the carboxyl group, and that hydrogen around the alpha carbon. So that doesn't differ. What differs among the amino acids are the R groups. So let's take a closer look at those. Let's practice labeling the parts of some amino acids. At the center, I've already identified the central alpha carbon. It's circled in red. Now attached to the alpha carbon, you're going to find the other groups. So here we have the carboxyl. Here's a carboxyl. Here's a carboxyl. Now that carboxyl may look a little funny to you, and that's probably because it has a negative symbol here instead of an H. Remember that carboxyl groups are acids, so what this molecule has done is it's ditched its hydrogen. It's put it out there, which is why it's classified as an acid. Next, we need to identify the amine groups. Amines are NH2 or NH3. In this case, they are NH3's pluses, and here they are in blue. Now amines act as bases, and so that's why they have taken that hydrogen out of solution and attached it to themselves. Now what's left over? Well, we have the R group. Everything from the alpha carbon down is going to be part of the R group. Notice that the R groups of these three amino acids are really different from one another. This is what gives them their different properties. The 20 different amino acids are classified in three categories. Some of them are nonpolar or hydrophobic, some are polar or hydrophilic, and some are electrically charged, and they can be broken down into either acids or bases. Let's take a look at them. Here we have the 20 different amino acids in their three categories. This top category is known as the nonpolar group. So why is that? Well, if you look at the tip end of the R groups of these amino acids, you'll see that they end in nonpolar functional groups, such as methyls or carbon rings. These amino acids do not like to interact with water. Here we have the polar group. Notice that the ends of these end in hydroxyls and things that like to interact with water. They are hydrophilic because water is polar and is hydrophilic. Here we have the acids and bases. Notice that the ones that are classified as acids end in carboxyls, which is an acid. The ones that are classified as bases end in amines, which are basic. Now, why do we care about these categories? Well, we're going to use these amino acids to build a larger protein. And when that protein begins to take on its shape, certain amino acids will only interact with certain other amino acids. So the amino acids that are used to build the protein are going to affect the shape that it eventually takes on. In general, nonpolars only like to hang out with nonpolars. They do not like charges. Polars, on the other hand, will bond with polars. They will sometimes bond with these electrically charged guys. And the electrically charged guys will often make bonds with any amino acid that has an opposite charge. Proteins don't exist as just long strings of amino acids. They have these complex three-dimensional shapes. So how do they achieve this? Well, that long string of amino acids has to fold on itself. So there are actually 3 to 4 different levels of folding that string has to go through to achieve this. Let's take a look at each one. The primary level of complexity consists of a long string of amino acids held together with peptide bonds. Now the sequence of these amino acids is really important. Because there are 20 different amino acids, any amino acid can appear at any particular position, but again they fall in a particular sequence. The sequence in which they fall is going to determine how this protein can begin folding and the shape it will eventually take. And the shape it eventually takes will determine its function. Where does that sequence come from? Well, it is coded for in the DNA. It is the DNA molecule that holds the instructions that tells the protein which amino acids to put in which order to build its primary structure. At the secondary level, that long string of amino acids, that long chain of pearls, is going to begin to fold on itself, and little shapes are going to begin to emerge. Now, what's actually bonding here? Well, it's actually the backbone elements of the amino acids that are going to bond. So it's the things that all the amino acids have in common, the carboxyl groups and the amine groups. What kind of bonds are they going to make? Well, look closely at the diagram. Dotted lines mean hydrogen bonds. Hydrogen bonds hold these little shapes together. Little curlicues begin to form, which we call an alpha helix, and that should be an alpha, not just an A. Or sometimes they form these kind of folded sheets known as beta pleated sheets, so that should be a beta, not just a B. That little chain of pearls that was beginning to fold up into secondary structures is now going to fold over on itself again and form what we know as the tertiary structure of the protein. At the secondary level, it was the backbone elements that were interacting with each other and making hydrogen bonds. At the tertiary level, we're now going to get the R groups involved. These R groups are going to reach across the protein and they're going to make bonds with one another, and that's going to twist that protein into its three-dimensional shape. Because there are many different R groups, there are many different kinds of bonds that can form at this level. Here we have an ionic bond. Here we have a disulfide bridge between two sulfur atoms. Here we have a hydrogen bond. It just depends on which R groups are present and their position as to which kinds of bonds will form. There is a special kind of interaction that we need to discuss at this level, known as a hydrophobic interaction. Now remember that some of the R groups are nonpolar or hydrophobic. Also remember that these proteins are going to exist in an aqueous environment, inside the watery environment of a cell. So when you have a protein like that that's forming and it's taking on a three-dimensional shape, and when it exists in water, it's going to fold so that its nonpolar regions are towards the center. In other words, this thing will twist so that its nonpolar R groups are pointed towards the middle, away from the water. We call this a hydrophobic interaction. You can see one right here between these methyl groups at the end of these R groups. Now, some polymers are done folding at this point. They only go to the tertiary level. If a protein can do its job at the tertiary level all by itself, we call it a fully formed protein, and there's no need for it to go on to the quaternary level. However, if that little subunit needs to get together with other subunits to form a bigger machine to do a different function, then it does need to go up one more level in complexity. We call these kinds of units polypeptide units. When two or more polypeptide subunits get together to form a larger machine, we say it has achieved the quaternary structure or the quaternary level of complexity. Here we have a protein that consists of two polypeptides making a bond with one another. Now it can be any number of types of bonds. Here we have hemoglobin. If you look closely, you'll notice that hemoglobin, which carries oxygen in your blood, is actually made of 4 subunits. And these 4 polypeptide subunits have to bond with each other in order for this thing to effectively carry oxygen in your blood. Now, why do proteins fold? Well, among proteins, structure is directly related to function. In other words, the three-dimensional shape that a protein takes on is directly related to the job it can do. This is especially important for proteins that have what's known as an active site. Here we have a three-dimensional model of a protein, and you can see that it kind of has this little cave in the side. It has a dent. That dent is known as the active site. Into this dent, a molecule known as a substrate will fit, and it will fit like a lock and key. And that's going to allow the protein to do something to that substrate, to break it or build it or move it or do something. If the shape of the active site is affected by a mutation or by damage, the substrate can't fit in there. The active site is ineffective, and the protein can't do its job. So among proteins, structure directly relates to function. Here we have 4 different proteins, each with different structures and different functions. The first is insulin. Now, insulin is a messenger. It is a hormone molecule. It's released by the pancreas when your blood sugar levels begin to rise. And so it will go out into the blood and it will travel around until it reaches the liver. It will bind with receptors on the liver cells and it will tell the liver to remove some of the sugar from your blood. The shape of this insulin is really important because if the shape isn't correct, it can't bind with the receptors on the liver cells, so it can't send its message. Here we have fibrin. Fibrin is used during times of injury, when you cut yourself, for instance. It's shaped like long strings, and what this will do is it will form an internal net across a cut. And if you look closely, you can see that there are red blood cells trapped in this net. So it kind of stops the bleeding at a cut until the coagulants and other clotting factors can kick in and make a proper blood clot. Here we have hemoglobin. Hemoglobin carries oxygen in your blood. To do this, it has to bind to iron molecules, and in order to do that, it has to have this particular shape. Again, it has 4 subunits, all of which hook together. Here again we have collagen. I mentioned it before. Collagen is shaped like a bundle of springs, and it's found in the deeper layers of your skin. It's what allows your skin to remain springy, to stretch, and to pop back into place. So 4 different proteins, 4 different structures, 4 different jobs. These things we call mutations can affect how a protein functions. Sickle cell anemia is an excellent example of a mutation that affects a protein. So to understand this, we have to understand hemoglobin. Here's the shape of a normal hemoglobin with its 4 subunits put together. Hemoglobin exists inside red blood cells, and because hemoglobin has this particular shape, red blood cells have this kind of a disc shape. Because hemoglobin is a protein, it's made of a string of amino acids, and they have to go in a certain sequence in order to fold up properly. In people with sickle cell anemia, a single letter on the DNA is changed. That results in a single amino acid being put in the sequence incorrectly. Look what that does to the shape. When this amino acid sequence folds up, it folds into this kind of weird shape. And inside the red blood cell, it sickles it. It pulls on that red blood cell and makes it kind of a crescent moon shape. Now, sickled cells like this don't carry oxygen as efficiently as the regular red blood cells. They also tend to kind of pile up and get stuck in the capillaries, so they can cause a lot of swelling and a lot of pain, especially in the joints. So this is an example of how a change in a single amino acid can have a big physical effect on the protein and on the cells in which that protein is found. Now, if proteins can be folded into a 3-dimensional shape, they can be unfolded. We refer to this action as denaturing. Denaturing occurs because of an exposure to heat or strong acids or bases. And exposure to these things breaks down the bonds that hold together the tertiary and secondary structures. So that protein is broken back down into its ribbon-like shape. Now, one of the major bonds that's broken during denaturing is a disulfide bond. That occurs between 2 sulfur atoms in the R groups of the amino acids. The effect of this is that sulfur is often released from a denaturing protein. So sometimes when proteins are denaturing, they smell little like boiled eggs. In fact, that's exactly what happens to eggs as you boil them. You may have noticed after you boil eggs that there's kind of a ring around the yolk. Sometimes it's kind of a greeny looking ring around in this area. Well, that is sulfur that is building up inside that egg from the denaturing of the proteins. And because there's a shell on the egg, it gets kind of trapped around the yolk. It doesn't hurt you to eat that. at all. It just looks kind of weird. Another place you might find denaturing is with hair permanents. If you've ever permed your hair and noticed that it smells like sulfur, well, that's because you're actually breaking down the proteins in your hair and reforming them into a different shape. And so sulfur is being released from your hair as a product of that. Denaturing is also a problem in cases of high sustained fever. If you get sick and you have a really high fever for a long time, the proteins in your blood and in your brain can begin to denature. That can cause organ failure. That's the reason that doctors will often try to bring a fever down by dunking someone in an ice bath or putting ice packs on the body, is to kind of break that cycle and prevent that denaturing. So denaturing is a powerful tool. It's also something you have to watch out for when you're sick. Hooray, you guys, you're finished! Arnold would be proud of you. You are now ready to attend class and put into practice all these great protein skills you've built up through this lecture. We'll see you then.